Welcome everyone. So in this video, I am going to talk about my team's bio AI activity for health technology. This lecture was given as a guest lecture to a course called Computer Science for Health Technology at UIT. I am re-recording this lecture so that people who want to know what kind of bio AI activity happening in bio AI lab at UIT Department of Computer Science. So my name is Dilip K. Prasad. I am an associate professor at Department of Computer Science, UIT, the Arctic University of Norway. So this campus is located in Tromsø, and dilip.prasad@uit.no is my email ID if you want to reach us. So who am I and about me? So I did my bachelor from Indian Institute of Technology, ISM Dhanbad in 2003 in computer science. And after that, I worked for 5 years in various companies like IBM, Philips, Panasonic, MediaTek, Infosys. And among them, Philips Singapore and Panasonic Singapore was in Singapore. I did various roles like software engineer, senior software engineer, and then finally I was a software architect at Philips Singapore. After that, I wanted more challenge in my life. I did my PhD in AI, ML, and computer vision topic from Nanyang Technological University, Singapore, 2018 to 2012, and I defended my thesis in 2013. After that, I was a researcher, postdoctoral researcher, research fellow, senior research fellow at NUS NTU for 6 years. And since Feb 2019, I joined UIT Department of Computer Science as an associate professor. And I have formed a bio AI group at the Department of Computer Science along with Professor Alexander Hosch. So in our group, we are 2 PIs, me and Professor Alexander Hosch. We have 5 PhD students and 2 master students, and we are going to recruit 2 PhD and 3 postdocs by February 2022. So what does bio AI means? So bio AI means biological signals and applying artificial intelligence to solve various kind of problem which can be helpful if we get a solution for the society. Like biological signal can be from the wearable device, it can be from EEG, it can be ECG, any kind of activity which a living being is generating. It can be at the organism level, like humans or animals, rat, cat, dog, or it can be an organ like heart, liver, kidney. It can be a tissue from different organ, and it can be a cell from a tissue. And using AI to solve various kind of problem or task which can help the society. So as I said, we are working in across the domain, like organism to organ to tissue to cell. Like wearable device generate various kind of signals like accelerometer data or heart rate data or electrodermal activity data. There are clinic and ICU machine which is generating smart ventilation data. We are also having a smart ventilator project using AI, then a gesture recognition using AI. And then if we go to organ level activity, that is EEG activity from the brain signals, and then from the different kind of scanning device like diagnostic imaging like PET scan, the MRI scan, the OCT, all this, and then the biopsies like histopathological data or different kind of tissue sample data, and finally at the cell and tissue level using microscopy and nanoscopy. All this signal, we are applying AI of various type like shallow learning, deep learning, using computer vision. Quite many data we have is image and video, then fuzzy logic, then we are doing time series analysis, we are generating simulated environment for AI to solve biological signal related problem. We are doing non-supervised learning, then using saliency. And combining all this is actually a bio-AI group activity. So in this video, I'm not going to cover about everything what we are doing in a bio-AI group. We are publishing many articles, but in this video, we are only going to cover three research area, which we have been doing in last 2.5 years. So it can be from the computer vision based medical diagnosis, then psychological health like motivation and sentiment or suicide, feeling in the human mind, then microscopy using physics-based artificial intelligence to help microscopists to know more about cell and tissue. Apart from these three activities, we do work with machine learning architecture like interpretable or explainable AI. Then we are building up scalable artificial intelligence architecture. Then evolutionary optimization, which are bio-inspired like ant colony, bee strike, genetic algorithm, the swarm. And we also doing this AI simulator using geometry and motion, light simulator, and we are also generating a dynamic complex environment where drone-based swarm like this can be helpful in generating a dynamic complex environment where we can develop the interpretable AI and scalable AI, and we have a proper ground truth to do it. So going back to our point, like in this, we are going to discuss about three topics, and let's move ahead with it. So first thing was like light OCT based deep learning network. So we had collaborated with All India Institute of Medical Science and Indian Institute of Technology, Delhi. And Ankit Butula, he's the first author of this paper. He is doing a PhD in IIT Delhi, and he was a visiting researcher in UIT. So he had no idea about machine learning or AI. So he wanted to get support or get more guidance on knowing AI and machine learning and apply for his kind of problem. So he had developed this OCT system, and that system was taking the different biological sample data. So data he was taking was the breast tissue data, and whether a person has a cancer or not cancer or any other disease from the breast tissue. So we started working on it, and then what we found that in this present world of deep learning architecture, people are going deeper and deeper and deeper and making model more complex. What we found in this, that instead of going deeper, we can go shallow and we can make problem less complex. And this is an example of the system where a sample is kept here, and then it is from the various things it get imaged. And if you see the OCT image of a breast tissue, the normal will look like this, and the tissue with the cancer disease at some places will look like this. In this case, both the false positive and false negatives are problematic. So what is a false positive means? So false positive means that someone don't have a cancer or a disease, and my AI system says or diagnosis says that a person has a cancer. Then the person will be scared. But if my system says false negative, that means if someone had a cancer and people come to know that it is the system says that the person don't have it, then it will get undetected, and this disease, which if it have been detected in early stage, will go to the advanced stage, and then the recovery of the person will be very problematic, and it may lead to a death of the person. Now, in the medical domain, what the doctors have to do, that they have to then and there take some tissue sample and see in the OCT, And then from this image, they have to visually, with their expertise, say that a person has this cell tissue is a normal or not normal, whether we should be removing that the whole tissue portion from that particular organ or not. Now, in the stressful condition like operation theater, where you have opened the body and then you are trying to find out whether this particular tissue area we should be removing, it has still got more spread of the disease or not. That is a very challenging task, and it is really stressful. So we come up with this AI-based diagnosis technique, which can actually help the doctors ease their task. And we have developed this very lightweight system, which can go into any kind of imaging device, which it has a very small footprint, very small memory area and computation need. So let's see. So in our light OCT architecture, is basically a two convolution layer is there. So one layer and then the second layer. And then there is a one fully connected layer is there. And with this small architecture and the number of kernel size we found is 5 is sufficient. So what was happening if we were going for too deep network like 100 layers or 1000 layers or 50 layers, the overfitting was happening. The reason of overfitting is like we have too few features. So network becoming sensitive by overtraining it. You can see the data is really, the contrast in the data is really low. The variation between different kind of gray is very low. And using this data, if you are training a deep network, it was causing an overfitting and the performance was really deteriorating. So then we design a shallow architecture where we only have two convolution layer and one fully connected layer. And with this very small footprint, we are able to give a state of the art, and the number of kernel required is only 5. And the performance of this system is around 99.7, 99.8%, and a deep layer network will give a similar performance. And another thing, very good thing we got in our light OCT architecture, like people want to know explainability of their model. So one of the explainability option they say is that this CAM, the activation map. So this is a second last layer, means last convolution layer in the Inception V3 model is a 94th layer. And if you see the normal cancerous image, the activation is here and the normal is activation is here. It does giving activation, and the performance of Inception V3 is also 99.7, 99.8%. And but if we see the second convolution layer of the Inception V3, we don't see much activation there. But if we see the second convolution layer, because we only have two convolution layer, and that is the layer, the last convolution layer is been used to find the explainability. As we can see that the cancerous image has activation like this, and normal tissue has activation everywhere. And this helped that the to medical specialists that it has an explainability, and also it brings the highlight that the explainability, people think like activation map is the one which can explain, but in our case, in this particular kind of sensitive data environment, the activation map is not able to really tell that what is it. It is explainable. It is giving some activity, but it is not actually explaining it. So putting this in the mind like activation map is explainability is not really a right in every places. So this was the task we did, light OCT architecture, very lightweight deep learning architecture we made. And it has a small footprint and it can go inside any system. And it has a very good performance for this OCT environment. The next task, next problem we will be seeing is that, which our group is doing, is the biological, psychological health activity. This work is done by a bachelor student who is a research interning with us from Jadavpur University. And Then this Laila Zari was an exchange student who actually done this design of the experiment and the EEG extraction of the data. And Soham, he actually worked on the AI solution for detecting motivation of it. And this student is able to do this work is for the very first time is able to do a motivation detection using EEG. So earlier there is no computational technique which does EEG which does motivation detection. The motivation detections are done based on the questionnaire by the psychologists and people who are working in social sciences. In computer science, this is the first work which actually trying to attempt motivation in the people using the EEG, how to find motivation from the EEG data. So we have taken a game-based approach. Our approach is basically a game-based. So what the game was designed like this, it is a memory game. Basically, it's a card game is there. And in the card game, the people, the users has to actually click at one card and then click at the other card. And if it is a match, it is going to see pair match. And it is going to say it is a pair match. And based on this pair match, the point will be given. And the users are being put on with the EEG data. And we have taken the 19 channel data of the users. And we designed the game both on a different way. Like there was a control group who had been allowed to play this game. And to them, no feedback was given, like it was a match or it was a not match. They can see it is a match, but there is no score increase or a match or unmatched feedback is given to them. And there was no point was given to them and no scores was given to them. And it is a control group. And then the next group, what we give that in the first session, we given them only the positive points. Like whenever they are doing a match, they are getting a positive points. And when they are doing a no match, negative points we were not giving. But in the next, after 2 minutes of break, they are being allowed to play the game again. And the next session, we are only giving, giving them the negative if they are doing a mistake. And in the third group, in the first, we are giving them the negative, and later on, we are giving them the score for positive. So when we are giving the score for positivity, we are not giving them negative. And when we are giving the punishing, we are giving the positive score as well as the negative score. So to know that whether it affects the human motivation, if they are getting the positive and they are getting the negative, or they are getting the negative or they are getting the positive. So this has been designed. And you see in this brain, this is the nose area and this is the ear area. And these are the 19 channel in the center. And you can see this is a reference point AFG, and then the another reference ground point is CPG. With this condition, we have taken the data. We did evaluate it whether persons are motivated or not, and we took the consumption of the data, and we see that when people are motivated, their performance means correct match of the cards is increasing with the time. That means they are able to memory recall and learn and keep in their short-term memory. This is a game designed for a use of short-term memory. And there is a clear separation in the motivated and not motivated. And when people are not motivated, doesn't really improve or disprove with the time. So this was a check to do a hypothesis check of our experiment, whether our hypothesis like game-based motivation we can detect or not. After that, we just did evaluate it, like we had only 10 participant in each of the group, like 10 in the non-treatment session, 10 in the punishing game session, and 10 in the rewarding game session. In order to validate our motivation and non-motivation, we had 10 participants in three groups. So in a control group 10 and the punishing game group 10, rewarding game group 10. As we see, the motivation of student, the number of users who were more motivated were 7 when a positive reward is given. And when the negative reward is given, some students were motivated even in the negative. For some students, the punishing, they like to perform better if they both positive and negative reward is there. But if there is no feedback or no reward is given or no punishment is given, usually the students, users are not motivated. And this actually very good, means it is a very small sample to say this, but it does indicate like giving a positive reward or positive feedback can help a better motivation in the student in the classroom. So if teachers give positive feedback, they motivate student better. Like rather than saying you did a 2-question mistake, and it's better to say that yes, you did attempted the 2-question and it was a good try. So this kind of feedback, this small study does say that giving a positive reward will help the motivation among the student. And this study was designed actually to understand the motivation. Like if students are better motivated, if the course design, the course contents are designed to have optimal EEG activity in the brain, means when the students are motivated and optimally activated, the learning will be better. So this project was started with that focus, and the motivation detection came from that if students are motivated, then we can test. If we can detect motivation, then we can test various educational course design work, various educational feedback system, or different kind of educational activity in a course to understand whether it is actually giving student motivated or not motivated. Then our motivation detection approach does say that it is doing the job, right? And but now, can we detect using EEG? Means from the feedback in the previous slide, we saw that it is showing that motivated, the number of positively correct answers people are giving more. But can we detect using the EEG activity? So now we recorded the 19 channel signals, and the 19 channel signals, the neuroscientists classify them. So the brain releases the signal from 0.5 hertz. That means 2 signals every second to 35-40 hertz signal. So 40 hertz means 40 this kind of activity, 40 this kind of activity in 1 second. So this different part of the brain when releasing the signal, and when we convert it to the Fourier domain, we can see this kind of thing. And the scientists have already said that if you want to know whether a person is doing a problem solving, we should see a beta signal that is from 13 to 35 hertz. And if want to see whether the person is relaxed or reflecting itself, then it is from 8 to 13 hertz. And if a person is meditating or doing some creativity, then it is a theta signal. And if a person is having a deep sleep, then it is a delta signal. Since the game we design or the experiment we design when the user is awake and they are doing some kind of problem solving, or they are in a relaxed state, like they are trying to find a match between a card and card in every consecutive click. So we choose alpha and beta signal for our further analysis. Now, for one more thing was there, like we human have the left side and we have a right side, and this front portion is known as the cortex of the brain. And all this the analysis and activity happen from here. And the frontal symmetry index, if we take into account, so there are if a person is approaching motivation, then F3, F7 channel, and if person is trying to avoid, then avoidance motivation is in the F4 and F8 channel. And if person has a positive emotion, then it is a T7 channel. A person has a negative emotion, because if you get a reward or punishment, then your emotions are supposed to evoke. And so we thought that this F3, F4, F7, F8, and T7, T8, as per the neuroscientists have approximately say that this indicate that. So what is F3, F7, F8? If we go back to this channel description, so F3, F4, F7, F8, and then T8 and T7. So these are the channels which selected for our motivation detection using EEG activity. So you can see in the channel selection, we have taken the emotion, we have taken the motivation, and we have taken the frontal symmetry. Based on this concept, we try to select the channel and try to find which kind of channel is giving a better classification of motivation and non-motivation using the EEG signal. And AFG was a ground and CPG was a reference. What we found that we did tested with a classical approach like K nearest neighbor, support vector machine. A why classical approach? Like any method which is not using a deep learning kind of architecture. And deep learning kind of architecture came in 2012. Now K nearest neighbor, support vector machine are decade old, 20, 30 years old, 40 years old system. And they are unsupervised. Support vector machine is also unsupervised in some condition, and otherwise it is supervised in certain condition. And what we found that unsupervised methods are giving a better accuracy than the deep learning method, like long short-term memory, multi-layer perceptron, and bidirectional long short-term memory, gated recurrent unit. Now, and these are supervised learning method. Ideally, the supervised learning method and a deep learning method and advanced method should give a much better accuracy. It was not making sense to us that why this performance was low for much advanced and better. Now, what we found that the supervised method was not giving the more accuracy. So what we found that it was doing actually overfitting. The number of data was less, as sample size is less. So what was happening with the deep architecture, it was overfitting. So how to sort it or help further overcome this kind of small sample size and overfitting? So Soham did an interesting and very smart job. Like there is this residual resonate architecture is there, right? Resonate architecture is proposed by Cunningham, who is now a scientist in Microsoft. Now, he used a resonate, but resonate was giving a very low performance, and deep, overfitting was happening. Sample size is small. So what he redesigned the resonate, and he used the residual of residual network. Means instead of using one residue, he used the more residue, and he designed this one-dimensional residue network. And with this modification, the overfitting he was able to solve, and still with a small sample size, the performance he was able to increase. We were able to increase the performance. So residue of residue is basically having a skip connection. So he created this residual block, one residual block, and put many of them in the residual block. And it helped in the unit level. If you want to know, residual block is basically have one convolution layer, and it has another convolution layer, and in between the Bayes normalization, and then there is a rectifier linear unit. If you know the deep learning architecture, you will be able to understand what these are the basic thing of the normal CNN architecture. And with using residual CNN architecture, he was able to handle this overfitting and small size sample problem. And voila, now you can see the performance is shoot down to 89%. And it is now making sense, like in order to solve overfitting problem or a small sample problem, the residue in a CNN might help. And it is first time we are able to do motivation detection up to a scale of around 90% accuracy with even a small sample size. In the paper, if you go, we have done the test called Friedman Test, and we did evaluate this architecture on 2 other data sets of EEG for doing other tasks. And with the Friedman Test, it says that our RR-CNN architecture model is much robust than it is the robustness test using a statistical test called Friedman Test, and it is a robust. And it is not that we are getting this result for one architecture. It is behaving strongly in one data set. It is behaving strongly in every other data set. So far we did for 2 kind of activity we are doing in our BioAI group for health technology. Let's move on to the next part of this session. We did work on the microscopy data, and this work is actually using a physics-based machine learning to do the subcellular segmentation in the living cell. And this paper has been accepted in Nature Machine Intelligence 3 weeks ago. And this work, the first author is Arif Ahmed. He's a postdoc, and he's an AI and computer vision specialist. And the second author is Ida Aufstadt. She is a biologist. She has done the experiment, designed the cell experiment. And Gustav, he's a PhD student. He is also a biologist working on cardiovascular disease research. And Åsa Birnas, he's an associate professor at School of Medicine in the University of UIT, the Arctic University of Norway. Walpit Singh is a physicist. He works in the photonics and design of microscope. And Krishna Agarwal is a physicist who works in the, she's a microscopist, and she also do a computational microscopy and computational physics. By using that, he's trying to improve the microscopes. And me, Dilip Prasad, who is a AI person. Together, it is a multidisciplinary, interdisciplinary work, and together we try to solve this problem. So what we are doing here in our group or in the consortium is we are trying to study why or when a certain kind of event happen inside a cell which triggers a different kind of cardiovascular disease. So one of the disease can be heart attack, why heart attack happens and when it happens. So what we are trying to study is trying to find where is the mitochondria and where is the different organelles like vesicles and how they are interacting. So using the AI, we first try to find, can we automatically detect and segment where are the mitochondria and where are the different kind of vesicles and organelles. So if we go to a camera domain, we do have a Hubble telescope to see the space, and if more expensive is the telescope, we can see more detail. As we see, this is a Hubble telescope, it has much more detail. Now, if we take a telescope from a sports shop or some nearby shopping mall, and that telescope, if we use it, it is not an expensive and doesn't have much detail like Hubble telescope. So the mean is that more expensive is, the more better is. Now, we come to camera. Like camera is designed the way humans see it. It is not designed to actually capture something more than what human can see. So it is designed in a such a way, the way human can see. So camera is doing exactly what we see. But when we go to microscope, it also has camera, but with a less expensive microscope, the amount of detail we see is not much. But if we go and see high expensive microscope, we can see more detail inside the microscope cell. So this is we can see more detail in the cell. But if we have a low cost microscope, we can maximum see cell nucleus and a cytoplasm. We will not be able to see inside the cell. Now, what further is there problematic in a microscope? Like if this is one molecule and this when go to a microscope, every microscope has this optical blur function and it blurs the data because this is called a point spread function. It's called a point spread function, and this is going to blur the data. And then this data, when it comes to the image sensor, and sensors are actually digital, it will further add an error to it. So when we see these 4 dots, this kind of thing, and this is a resolution like 200 nanometer, and when it goes to the camera from the microscope, we will not be able to see 4 things. It will be seen as one item. Now, why we are discussing this? See, inside the cell, when we have an organelle like mitochondria and vesicles, now mitochondria are energy courier, like heart, our human heart. We are studying a cardiovascular disease, right? So it is related to human heart. Now, human heart beats constantly for the whole life. Even the moment it starts beating at 6th week in the mother's womb, and from there until the death, it is keep on beating. Now, how does it beat? It needs energy to beat, right? So mitochondria is energy courier inside the heart cell, and mitochondria are there in every other cell in our body. And the size of the mitochondria will be around 200 to 500 nanometer. And while the other small organelle like vesicles, it will be 50 nanometer to 200 nanometer. Now, a normal microscope, even if it is an advanced microscope, it is not a super resolution microscope. It can see up to 300 nanometer if a particular wavelength like laser has been used, like 600 nanometer laser if had been used, which is in a visible spectrum. Then we can see up to 300 nanometer. And if we can understand how mitochondria and how vesicles are interacting, if some kind of experiment is done, we will be able to know what is happening inside the cell. Now, example, you see during the corona time, it took scientists, imagine if we scientists know that what is happening inside the cell by looking at the video, then they could easily find out that why this corona is affecting and how this corona is affecting a cell. And if they understand how it is happening in the cell, they can tell what problem is happening. And if they are developing a drug to actually say whether it can treat the human body or not. So by using that chemical in the cell, it can find out that whether that particular drug is helping in a desired way to overcome a disease or not. Now, so far, we don't have a microscope which can do a live imaging inside the cell without altering the cell. And we are working towards that direction to make microscopes a knowledge microscope rather than the imaging microscope. So mitochondria are basically like a tube-like structures, and on the micro tube-like structure, the chemical agents are added. Those are called fluorophore. And that fluorophores are actually imaged by the camera. So at the end, we just see the fluorophore. We don't see anything inside it. So as a result, what we see is basically a thread-like structures. Mitochondria will appear like a thread-like structures, and we don't see it as a tube-like structure. And that is a bigger, another challenge also. So this particular is generated by the Blender software and is a simulation, but this right side is a real data. what we see inside the cell, and it has been tagged with different colors. Now, why we are doing this physics-based simulation? Why not? Now, if we go here and if we ask scientists, if you want to apply AI, the best currently available we have is a deep learning approach. Everyone say apply deep learning. Yes, deep learning is doing a very good job. Now, a deep learning is going to, for deep learning, you need a lot of data to be given to people, right? You need a lot of data to be given to people. If we know inside the cell the detail, then finding the drugs, whether they are effective or not effective, will be easy. Now, the thing is that the deep learning architectures are there to do the task, but the deep learning architecture or machine learning will need a lot of annotation, pixel by pixel. Now, if we go and see, even if there are 4 this kind of dots are there, and to the camera it will appear like this, and if we add a noise, then it will be something else. So practically, there is a resolution limit of the camera, and if something is what we are looking is 200 nanometer and 50 nanometer, and even the best the microscope which are used, and they have a 1 pixel resolution of 80 nanometer. That means if any scientist is trying to mark pixel by pixel, and he or she does 1 pixel error, then if it was 100 nanometer, it will be 180 nanometer. So instead of if it was a 200 nanometer, it will be a 280 nanometer. So instead of vesicle, it will be appear as a mitochondria. And if he does mistake, then instead of mitochondria, it will be classified as a vesicle. So, and that's what we saw result. We given a very simple data to different kind of scientists, like a life scientist, microscopists, data scientists. And you can see this is a simulation data generated using our simulation engine for microscopy, for mitochondria generation. And this simple data in ground truth will be like this, but even the microscopist who's supposed to know the microscope or a life scientist, they have done error. They have done error. They are not able to detect. So the red one is saying incorrect annotation, and the blue one, the cyan one, is saying the correct annotation. So you can see even with the simple thing, the similarity match is 77% to 46%. So what to take? We can employ, you can put many life scientists or microscopists to do the annotation, and then take the mean of them. So if we take mean of them, then 46% match is there. And if we take the median of them, then 46% match is there. And if we take whatever correct, even a single pixel correctly put, because we know that this is a correct one, then also the 77% will be the best possible accuracy we can go for this particular image. Or 81% even taking the every correct. Now, as a correct, every correct pixel, even single voted as a single time, if we take it as a correct detection, then also we can go up to 81% match. Now, this is a very small data. And if we want to see how much time it takes to annotate for this particular image, is 10 seconds. It takes 10 seconds to do one. Now imagine this is a real cell experiment data. It has a 2 cell. This is a 1 nucleus, this is another nucleus. So to do annotation of each and every mitochondria for one image, it will take 3 hours per cell. Now, if we do a manual segmentation annotation to give for deep learning, it will take days. And if we take unsupervised, it can do second, and we will see in the next slide what is the result of them. And if we do simulation supervised, it will also take second. Now, once we have developed the model, we are able to generate a lot of simulation data. Now, for deep learning, we need around 5,000 to 6,000 images, or even 1,000 images of different variety. And out of 1,000, you have to do the biological experiment. Now, biological experiment, to do it, it is not like clicking a camera of a scene. You have to prepare the sample, you have to add chemical, you have to subtract chemical, you have to wash it, then a lot of process you have to follow, then you are able to get an image. And it is a reality that even if you do 5 different experiment of the cell, you are able to successfully get this kind of good data 2 or 3 times. So to get 1,000 different experiment, it is going to take years, and then you have to put annotation for years. This is why this is going to be finding out each and every annotated data and every mitochondria in the cell. First of all, it is erroneous. Second, it is taking a lot of time. So this is not a right way. So we went to the approach like we know how the physics is, we know how the microscope generate the data, and we know how sensor get the data, and we know AI. So we come together to solve this problem, that can we do this training using the simulation supervision engine, rather than asking the scientists to put their time for years to do the annotation. As I was saying, the unsupervised technique. So now we see unsupervised method. So these are the results of unsupervised method, and this is a ground truth given by the life scientist. And this is the real data from the cell, one small snapshot of one small portion of the cell. So many mitochondria are there. Also is a very well-known method which does annotation of it. And then there is an image is there, which is also has a tool. If you see our paper, you will come to know which method we are talking about. And then this adaptive method is there, which does give this kind of result. There is a method, backpropagation, also available, that gives 62% accuracy as per the SSIM score. These are all unsupervised method, can do a job in a few seconds. But do you really think that this data has any way match with this data? We want this kind of segmentation, not this kind of segmentation. So we need a better job. So what we did, the usual way that user expert do the annotation of it and then pass to the training data, and then after the training apply to the real data, and then you get the segmentation. We took this approach. We did physics-based simulation. We simulated the data, and since we simulated the data, we know what is a physical ground truth, and that is a correct ground truth based on the physics concept. So physics will not lie. And then apply this machine learning training, and then after the training apply to the live cell, and we get this kind of segmentation result. So that is our approach. And how we do it? Basically, we want to model the mitochondria. We have to 3D model them, how the mitochondria are actually in length and breadth and size and shapes. And once we model it, and then we have to put emitters, like how the fluorescent labels are getting tagged, and their estimate, and also their photokinetics from the manufacturer. Like if we are using M-cherry, what is the emission spectrum of the M-cherry, and how much photon they release. So counting each and every photon, and that integrated in our physics-based simulation, and then apply the camera blurring function called point spread function. And then we get the convolved image. Now, normal microscopy data, we do have noise. So we also added a noise which is a realistic in a particular microscope environment. And after that, we get this binarized data. So this is the ground truth we get. And this is used for training. And after the training, we trained the various deep learning methods. We did train the semantic, fixed-to-fixed, deep CNN, master CNN, UNet. All these different kind of method we actually did the training, and we found that these trained deep learning methods are giving much better accuracy for real data. Our proposition is that we are getting a better result than what actually we get, because this segmentation is done by the scientists and it is limited by the resolution accuracy. So resolution of the camera is not what human can see. And also we did saw that scientists do fail. No one is able to detect correctly. It is very difficult to do 1.00 here, even such a simple. Now, when we go for this kind of complex, it is very difficult for anyone to tell exact pixel by pixel match. So we do getting this better accuracy. But actually, if we have a proper correct ground truth verified, then this accuracy will go higher. And in simulation environment, we are getting above 98% accuracy in our experiment. So we see physics-based simulation supervised learning does work. And with this, what is a flexibility what we are giving? The flexibility which we are giving is that once we have modeled it, right? If you want to if you have a new microscope, if someone has a new microscope and with that they are capturing the data, we in our model, in the simulation environment, we just change the parameter of the microscope, matching with that microscope. We generate the simulation data and retrain it. If we have a new structure, we did it for mitochondria, vesicle. If we want to do for endoplasmic reticulum, we model that cell organelle. And then we can generate the data. Now, if we get a new chemical like EGFP, M-cherry, these are the fluorescent tagging label. There are over 200, 300 of fluorescent taggers. And if we get if we have our experiment condition has a demand that one should use this kind of fluorescent labels, then we just change the parameter of fluorescent labels and regenerate the training data and retrain it. Now a new kind of method has came up, and this method is giving a better result. So we just change the model. As we can see that it does work for so many models. A new model, if we just train it, then it will work. So it is giving a lot of flexibility in the system, and it is applicable and useful for many different kind of experiment related to live cell organelle segmentation. So what we got output, and this is a result of real data. As we see, you can see this. This is the mitochondria has been identified with identification number is given. And the green is a single rod mitochondria, and the small size is a dot mitochondria. And if it is a cyan color, is a network of mitochondria. So if we can identify and track them, as we saw that they are moving, we can tell that what it did here. And after that, it moved away. So what happened after a few seconds to this organelle, whether they combine with each other or they move somewhere or this came. So this kind of event and model we can find out, and we know really what is happening inside the cell, why a particular disease happened, why a particular drug is working and not working. Currently, we have to go for clinical trial where you administer in the animal, we do the lab experiment, and then we go for a clinical trial. And then clinical trial, we only getting the blood sample and finding some parameter from the blood. We really not looking inside a person's body or a cell for which the drugs are getting developed. And that is a black box testing is getting done, and it taking 7-8 years. Now, if you know anything, we have the technology which can really see inside the cell. Then finding a disease cause or finding a drug is working or not working, that's the time period can be greatly reduced. We don't take years. We will precisely know what is actually doing that drug inside the particular system. Now, if you we did for so this is our cell data, and on this the red one is actually vesicle tagged, and cell data actually look like this. And if you ask someone to tag which one is which one and tag each and every pixel correctly, it is a Herculean task. So by doing this, we are able to do this segmentation of or identification and segmentation of mitochondria and vesicle. The green one are mitochondria, red one are vesicle. So you see, we can know that what is happening, which organelle is going where, and when they interacted, what happened. As you see, the number 9 is going to this mitochondria, and it is going to touch it, and then it go here, and then it did some kind of activity here, and he stayed some time here, and then it move away. Now, if then we further evaluate what happened to this mitochondria, what chemical it releases, and what is the fate of it, we will be able to know that what happening inside and what is the root cause of particular disease or of any kind of thing happening inside the cell. Further, we can also know that what kind of motion they are doing, whether they are going straight or they are having a random motion. So this one you see is a vesicle. This is a small portion which we are zoomed in, and the red one is a vesicle and the green one is a mitochondria. And in the right, during our nanoscale motion pattern classification, we are able to actually classify in a very nano level. So imagine these are very small nanometer size organelle, and their motions are really very micro level motions are there. They're not doing much motion. But with our AI, we are able to classify what they are doing. Here they were stationary when they were touching the mitochondria. They might have exchanged some chemicals. Now, to know what chemical they have exchanged, we need a much higher resolution, or we have to apply a super resolution technique to find out what exchange had happened. So this is an ongoing task. So our core bio-AI topic has many interesting problems, and out of the box solution we seek, and anyone who is going to join our team, we will need someone who like to think out of the box to do this kind of activity. We have many other interesting problems, machine learning architecture like interpretable, scalable. We are generating AI simulators and doing a drone-based dynamic complex environment, modeling a particular cell, and doing evolutionary optimization. We have a team, our team is growing. We are going to be 14, 15 people team as a core team. And if we take our collaborator and consortium, we are a team of really 70, 80 people. If we take our collaborator. And currently we are doing research on what we are doing. Another interesting activity is that we are taking a cell from human skin, and we are applying a stem cell research. Our consortium partner like Osa Berna, who is from School of Medicine, she is applying the pluripotent technique, a stem cell research, converting it to the cardiac cell, which is known as cardiac myocyte, and then putting that cell into a test tube, and then growing it and making it an engineered heart tissue. And you see the engineered heart tissue is actually beating. This is a real data of the lab who has made this engineered heart tissue. And this tissue we are imaging using the microscope, and after that we are applying AI to find out at the tissue level what interaction happened between a cell and what chemical are getting exchanged, what actually causing a particular problem. So a lot of interesting thing which we are doing it in a lot of young and mind. We need a very challenging people we need who like to see the challenges. And we are further doing to find out, develop the dynamic complex environment. We are having this experiment where we are having a swarm drone. Many drones are there, swarm of drones are there, and we are trying to get the experimental condition.
Wouldn't it be great if we could send in robots to find the gas leak quickly and without endangering human lives? We have developed the first autonomous swarm of tiny drones that is able to find gas leaks in unknown, cluttered environments. Their small size not only allows them to...
So in this experiment, what they have done, they are using this drone is to find out the gas leak in a place where human will have a health hazard if they go and find out. We are going to use this swarm to develop this dynamic complex environment, get the real ground truth data. You know, getting a real ground truth is hard. In a cell environment, getting a real XYZ position, even a best life scientist or microscopist will not be able to tell it because they are limited by what camera can see. So using this dynamic modeling, we can get XYZ and other parameters, which is verifiable. We are going to work further on it. So this is a brief about our thing. We are working on interpretability, explainability, scalability. And explainability means we are training this deep learning model with cat, and cat, what neural network, neurons, or nodes they are activating, and finally in the deep learning models are learning. We are trying to find how different deep learning models are able to explain it, or whether we can develop and both explainable and scalable AI technique in our team. So we need really a interesting team, interesting people, and who like to take challenges. We have a very sound challenge and a strong team with a dynamic team working on to solve the really hard problem of this AI technique in the bio AI domain. So our task is not done by one person. We have partners from the University of Hospital Hamburg, then the Cancer Research Center in Milan, then there's SME third place, University of Barcelona, then University Hospital Norway, and YenLab, SME. If you want to know more about our team, you can do this QR code and you can go to BioAI Lab. And thanks for listening, and shoot me if you have any further question. Thank you very much, and see you again. Bye-bye.